Okay, let's get started. All right, have you ever had a coach tell you last one, best one? Okay, that's what we got today. Last one. We're going to hit all of the major pandemics today. So of the 20th and 21st century. So we've got flu, we got HIV, we got coronavirus. Yeah, all right. So little business though. One more person in here. Little business, I did post some updated learning objectives. So I've had enough people ask me about it. Do we have to know this? We have to know that. I went through this last Monday in last Monday's lecture, like the new stuff that's going to be on the lecture. Sorry, the final. But again, I did post updated learning objectives with some things crossed out. So no whooping cough, no tetanus, no botulism. There's some other stuff like measles and equine encephalitis that we're not going to do. So if you have any question about that, again, the test has stuff crossed off. All right, so with all my technical difficulties at the beginning of the last lecture, I forgot to record the lecture. So remember how I said there's not enough hours in the day lately. So when I got home that night, I just put on the lectures that I recorded from last semester. So it's really pretty much exactly, I even made one of the same jokes in class. So I think that's really comparable. The one thing that got left off is that I talked about treatment of gonorrhea last lecture. If you just know that, hey, antibiotics for now are good to treat gonorrhea. We worry about the resistance there, but and that may be coming in a time in the near future, but for now, for the most part, antibiotics are good to treat that. In general, people are asking me also pointers to study for the final exam. Make sure you're looking at the new stuff, giving that your most amount of your attention and studying for the final on Monday. Remember, homework due on Monday as well. Try to be systematic. So you're looking at, virulence factors, diseases that the organism causes. What's the morphology? Is it a cocci? Is it a rod? Is it a DNA virus? Is it an RNA virus? In terms of treatment, if it's antibiotic treatment, just, antibiotics is good enough. For some of the viral infections, we're talking about some other drugs and how they work, and that is more fair game. Okay, so kind of strike a balance with the treatment. You don't have to know individual antibiotics, but in some cases, like for herpes virus, we talked about acyclovir. Okay, it's a nucleoside analog. All right, you should recognize that as being something that can be helpful with herpes infections, okay? All right, so we're starting our RNA viruses, it's chapter 25, the Ortho Mixoviridae. So that's the family name. There's lots of Ortho Mixoviruses in that family. Not all of them are just human. So there's about 5 different orthomyxoviruses that infect humans. The 3 types, especially that are famous, the 3 subtypes, of course, this causes influenza. I'll write that here. That'll be obvious. 3 subtypes of influenza. Influenza virus A, influenza virus B, and influenza virus C. All right, A is the one that's typically causes the most severe disease. And that will be a little bit more apparent in a minute. That's influenza A viruses can also infect animals. So we get some reassortment there and some novel things. So influenza A is typically more serious in humans, B a little bit milder. And C is usually pretty benign, maybe acts more like a cold in terms of symptoms. So that's not one that we worry about as much. Okay, characteristics of the virus, it's enveloped. Okay, so if you look at the little diagram over there. You can see that phospholipid membrane. Remember, the envelopes are, they're taking those membranes from their host cells, so they're going to be phospholipid membranes. And a segmented genome, and it's actually negative single-stranded RNA genome. All right, so you can see in this, again, this graphic to the right, you can see that the segments, an influenza virus will either have 7 or 8 segments. I think influenza A and B have, A has 8 and B and C have 7 segments, okay? The capsid structure is a little variable, so we're not going to worry about that. Each one of these segments encodes for either 1 or 2 proteins. So it's actually about 12 proteins that we think that genome encodes for. So that's a lot less than herpes virus. We said there was like 80 or 100. So smaller genome. And that single-stranded RNA, remember, it has to make the complementary strand to make positive sense RNA, and that's what's read at our host ribosomes. Okay, and then that positive sense is copied into the negative sense to make more virions. All right, the spikes. There's 2 types of glycoprotein spikes on the surface of the influenza virus. Remember, glyco, sugar, and protein. This is the part of the virion that kind of gets all the attention because that's the part that's the most antigenic. Okay, remember we talked about how proteins are good antigens. They're eliciting a stronger immune response. Okay, same thing we'll see in coronavirus. The spikes are what gets all the attention. All right, so there's 2 types of spikes. The H spike, that stands for hemagglutinin. Oops, only 1 T. Sorry, hemagglutinin. The hemagglutinin spikes there are responsible to help the virion attach. So attachment and fusion to the host cell. Okay, and the other glycoprotein spike, we call it N, that's Neuraminidase. Okay, you can see that over here. Neuraminidase is especially important for release. So release of the virion from that host cell. After many new virions have been made, okay, that something is necessary to kind of cleave off those new virions so they can release from the host cell. Okay, and there's actually 16 different types of hemagglutinin and 9 different types of neuraminidase. All right, so we use those different types to name our different strains of influenza virus. So for example, the strain nomenclature here, how we name viral strains, it looks pretty confusing, but if you pick this apart here, here's a couple of examples. For instance, A, these are both influenza A. Remember, those are ones that usually cause the most severe disease. And then sometimes it's included in the nomenclature where we originally isolated that virus from. You know, was it a horse? Was it a pig? Was it a duck? Whatever, human. Where we originally first, where it was first sequenced, I guess, or where it was sequenced from. This is a strain number. This was the year that it was isolated. And then here we have like H1N1, the first subtype of the hemagglutinin and the first subtype of neuraminidase. Okay, H1N1 is also known as swine flu. All right, so we are robustly following the different kinds of strains of flu that are circulating because those are the strains that we need to make the vaccine to work against. So a robust surveillance as to what strains are circulating it's very common for RNA viruses to change. All right, they mutate pretty rapidly. All right, so let's talk about that, the change ability and a little bit of how that works. All right, because our vaccine typically is only maybe 40 or 50% at best effective. Why is that? It's because of the antigenic variation that we see with the orthomyxovirus. All right, so we talked about antigenic variation with gonorrhea as well. Neisseria gonorrhoeae does a little different kind of antigenic variation that's very quick, like every 10 days or so it changes to stay ahead of our immune system. This is a much slower change, like over a period of months and, you know, kind of throughout populations, not kind of typically within one host. All right, so two kinds of antigenic variation in influenza. Antigenic drift is a very gradual change over time, point mutations in those spike proteins. So insertions, deletions, maybe substitutions that, you know, again, cause a very small changes over time. So we think antigenic drift is responsible for a kind of every other year increase in influenza, right? I've mentioned influenza is an endemic disease. Like we always have that at some kind of level in our population, but yeah, you know, if that kind of spikes up, we can have kind of outbreaks, or we could even consider it an epidemic. 10 years ago, 2008, 2009 was the last time we had a flu epidemic in this country. So, and if you look at the data, typically, again, every other year is kind of, you know, one year's not so bad, next year's worse, next year's not so bad. Okay, and we attribute that just to kind of slow antigenic drift. Antigenic shift, on the other hand, only type A influenza does this, because that's the only type that will cross species. Okay, and this is when we get 2 or more influenza viruses infecting a single cell. We get some major reassortments and end up with novel combinations of hemagglutinin and neuraminidase. All right, so I'll come back to this slide in a second, but this was the best graphic that I could find that kind of illustrated that antigenic shift. Okay, so for example, say a human influenza virus and an avian influenza virus both infect another organism, say a pig, all right? And you get some reassortments there, and you end up, the new virions being made have that human, it's part of the avian H3 and the human N2, and it's something that maybe is transmissible to humans. So this is something novel that people wouldn't have antibodies to, and it's more likely to cause you know, more severe disease. Okay, and then again, antigenic drift is just kind of slow point mutations. So we estimate that maybe every 10 years or so, we get kind of a big spike in flu due to some kind of antigenic shift. Right. So like I said, we're a little overdue now. It's been about 12 years ago since we had the last pandemic. I don't know if that was, you know, 10 years ago, you guys would have been pretty young, but at that time we had just, you'd hear of people dying of flu that were not otherwise in poor health. So that was a pretty rough year. And again, we're due for that. We were worried that that would happen this winter, and we were worried that the healthcare system couldn't take flu and coronavirus at the same time. Sounds like the masks really mitigated any threat that we would have of flu. So we're thankful for that. All right, so how is flu transmitted? I think that's probably not too hard. We've got large particle droplets. Okay, so sneezing, coughing, We think that's primarily the way the influenza is passed. So, you know, typically less than 1 meter. It has the potential to be airborne. Okay, if those viruses are in, you know, the small droplets that, you know, as we speak, again, as we cough, remember we consider airborne to be passed greater than 6 meters. That's potentially as well, but probably kind of like coronavirus, that's probably not the main way that it's transmitted. Okay, also can be transmitted by fomites, but like coronavirus, less often, okay, because this is a membrane virus. It's going to be very wimpy in the environment. It doesn't stay viable very long in the environment on some surface. So we think influenza may be viable up to a day, which is a really long time, apparently, for a membrane virus. Okay. Of course, in areas of high population density, there's going to be a lot more transition when people are packed in together. All right, so 100 years ago, little historical perspective, this was just the perfect storm for a pandemic. We had, you know, some novel strain show up, and because of World War I, troops were moving all over the world. We had, you know, army camps, people living in close contact with each other. It's estimated that between 1/3 and a half of the population of the world was infected, and about 5% mortality. All right, so that's somewhere in the area of 50 million people dead. So we're not there yet. That's an estimation, but with our current pandemic, thankfully, we're not there yet. A lot of people think, well, this is just the flu, right? How could this happen? How could people die of this? This was not a normal strain of flu. This was a strain that was actually most lethal for people in their prime. People ages 18 to 40 were the most common age group that was dying of influenza. So normally it's the elderly and the young that are most susceptible, probably because of their robust immune response. It was the inflammation, excessive inflammation, that was causing some of these deaths. But it had all kinds of crazy symptoms that you don't see normally with flu, like hemorrhaging. You know, people would bleed out their nose, bloody coughs, had neurological effects. When President Wilson got the flu, and he recovered from it, but he was different. Like his decision making was different after he had the flu, and that probably played a role into the treaty that was made after World War I. Was really tough on Germany, and all along he'd said he was going to, you know, be easy on Germany. So, and then he ended up dying like a month later of a stroke, which was probably brought on by the flu. So, really, really interesting to think about that. Was this pandemic caused by antigenic shift or antigenic drift? Our thinking for a long time was that there was some major shift that happened. And by the way, most of these flu pandemics we worry about now coming out of China, because in China you have people living sometimes in the same house with their pigs and their ducks and their chickens, like all together. So it's not surprising. We're always kind of especially monitoring China, worried about these novel combinations. But this is a question that I don't know. We're not going to jump into this right now. There is some studies that say it was more of a gradual drift into something that was kind of a novel combination. Okay. Clinical disease, I think you've probably all had the flu at some point. Let me stress that the flu is not vomiting. All right, so it's one of my pet peeves when somebody says, oh, I had the flu, I was throwing up all night. Okay, that's some kind of, let's at least call it a stomach bug, or at least call it the stomach flu, which is not a great description either. All right, influenza is respiratory. All right, so you're having coughing, muscle pain, high fever, chills. And this is lasting sometimes a week, sometimes more. It's really miserable when you get a good infection. Typically, about a 2-day incubation. I don't think people are typically contagious until they start showing symptoms, right? So that's always a huge problem when people are contagious when they don't have symptoms, as we know. All right, so it's killing host cells in the respiratory tract, lots of cytokine release, inflammation. Secondary infections are really common. So especially with young children, to have the flu turn into some kind of ear infection, their ear canals are really narrow. And for the elderly, it turning into some kind of bacterial pneumonia is a huge problem as well. With that 1918 pandemic, pneumonia was a huge problem. Of course, this was before antibiotics. They were pretty good at treating bacterial pneumonia back then. They would harvest antibodies from people that had recovered or from animals. When you have, things like this, gymnasiums full of cots of people that are sick, you don't have time to harvest antibodies to treat all those patients. So, the worry is that we're really not that much better at treating flu today than what we were 100 years ago. Let's talk about treatment. The main drugs that we use to treat flu are all considered neuraminidase inhibitors. All right, so this graphic I got off online, not in your book. Ignore this part of it over here. Amantadine is a drug that's not used anymore because all the flu strains are resistant to it. But over here, this NAIs, neuraminidase inhibitors, all right? So this is where they work. They inhibit the release of virions. So over here, this is what's supposed to happen. The virion buds off and releases, right? But the neuraminidase kind of keeps those virions stuck to the outside of the host cell. So that's going to slow down their replication. And if they do release, they tend to kind of clump together. And we know that our immune system is better at noticing big clumps of things. So that's, again, hopefully going to make it a little easier for our antibodies to find them or slow down replication. All right, so that's how neuraminidase inhibitors work. I just wrote the trade names here of the 3 drugs that are used now. Tamiflu is probably the one that's most familiar. Controversial whether these drugs really do that much good. There's been a lot of studies that have said that maybe they decrease the illness number of days that you're sick by about 1 day. All right, so if you're sick 6 days instead of 7, yeah, that's great. Not sure if that's really worth it or not. You also have to start taking them within 48 hours of your first symptoms. And for now, you need a prescription. So when you're feeling lousy, you got to drag yourself out of bed, possibly expose other people when you go to the doctor to get the prescription. That will probably change very soon, that you'll be able to get it over the counter. That's in the works, if not available already. And they're also not as expensive as they were because generic versions are coming out. So they're more like $20 or $30 rather than $100. Okay, so with any drug, there's potential side effects. So, nausea, headache are potential side effects, which is, right, you already got flu symptoms to deal with, so that's not good. And with any drug, there's potential allergic reactions. Anecdotal evidence, but I had a friend whose 2-year-old came down with a flu. The doctor prescribed Tamiflu. She had started having seizures. They figured out from the Tamiflu. So it's a conversation to have. Educate yourself. Conversation with your doctor whether it's it would be good to take the drug or not. They do recommend it for anybody that's immunocompromised, really in danger of having, you know, the bat flu really bad, or, you know, very severe flu. Okay, so it's a whole, like a whole discussion in itself. There is a new flu drug that came out in 2018 called Zoflooza. So you might have heard the commercials for that on TV, not so much this winter, but last winter. Flu really sucks the life out of you or something like that. Those are the Zoflooza commercials. This is a different mechanism. This inhibits cap snatching. All right, and I looked and looked for a good graphic of this and I couldn't find it, but this, we have talked about this. So remember during transcription, we talked about the difference between prokaryotic transcription and eukaryotic transcription. The eukaryotes put a 5' guanine cap on their transcripts and a 3' polyadenylation. Okay, because that transcript has to travel from the nucleus out into the cytoplasm, so it can protect the ends of that messenger RNA. And then the 5' cap is read at the ribosome, kind of lines up at the ribosome there. All right, so if viruses are going to take over our cellular machinery, take over our ribosomes, they actually have to acquire a 5' cap on their transcripts so that it can be read correctly at the ribosomes. Okay, so they do this thing called cap snatching. Okay, they snatch this 5' guanine bit there and add it on to their messenger RNA. So what Zoflooza does is inhibits that cap snatching so that it's less likely for the viral messenger RNA to get translated at the ribosome. All right, and the studies that I've looked at say that this works a little bit better than the neuraminidase inhibitors. It is still pretty costly. Let's see, did I write down how much it is? $150, $150 to $180. One pill though. You still have to take it, you know, right when you're, you know, within the first couple days. But that's kind of nice. You only have one dose there. Maybe a few less side effects, but only for people that are older than 12 years. So not giving this to kids yet. So maybe a little bit better, but you know, still some drawbacks there. Other than those two things, you know, rest, stay in bed, drink lots of fluids. You always, especially for kids, you don't give them aspirin because of the risk of that brain inflammation, Rise syndrome. So mostly, you know, Tylenol there is pretty much what we got. Okay. In terms of the vaccine, there are many different combinations of the flu vaccine. There's a lot of different ways you can take it. Most of the time, you know, somebody that's healthy, you know, not too old or not too young, they give an inactivated virus. It's injected. You can get it. Typically, it's grown in eggs, and then it's inactivated and purified after, you know, takes a number of months to grow up in the egg. So we actually have to decide what strains we're going to put in the flu vaccine. They met in March of this year to decide which strains will go into the formulation for next winter. So they need a lot of time to grow these up. If we could get, say, an RNA vaccine that worked for flu, then production, you know, would only maybe take a much shorter amount of time, and we could, you know, better get a better grasp of the strains that are circulating at that time. So hopefully that maybe that will happen in the near future. They have versions of this that are grown in cells, so that if you're allergic to eggs, you could still take it. Kids, they like to give kids the attenuated version of it. That's, you just kind of sniff it up in your nose. That actually works a little bit better for kids. So most years they offer it both ways. For elderly adults, they give a kind of beefed up version of it. And I just included here last year's formulation of the flu vaccine and this year's. Sometimes they include some of the same strains. They either, for a quadrivalent vaccine, they include 4 strains. Sometimes they give it in the form of a trivalent vaccine with only 3 strains, but they usually include a couple of A's and a couple of B's there. All right, so don't memorize those, just kind of, hey, this is kind of interesting, you know what some of that stuff means now, okay? So it is good to get your flu vaccine. I know people that don't like to do it because they get so sore and feel so lousy afterwards. First couple of years I got the flu vaccine, I felt like that. And now when I get it, it doesn't even make me sore this year. So kind of more times you get it, I think that's pretty common that your body doesn't react quite the same way to it. And, you know, over time being exposed to all those different strains, that if something new does show up, hopefully, you know, I will have some immunity to it because I've had so many flu shots over the years. Again, it's typically flu vaccines somewhere between 40% effective. So yeah, you can still get the flu, even if you've had a flu shot. You know, you could get a strain that's not included there. Usually your flu is going to be milder, you know, if you've had a flu shot. So I would encourage you to do that. All right, any questions about influenza? All right, let's move on to HIV. Okay, also an RNA virus. So the family here is Retroviridae, because all the viruses in that family make they're retroviruses. That means they make reverse transcriptase. So we're going to talk about that enzyme. Lentivirus, it's a little confusing because it's not kind of all the same name. Human immunodeficiency virus is considered a lentivirus. All right, so it's another enveloped virus. It has a segmented positive sense RNA. So here we've only got 2 segments. It has 2 copies of the same segment there inside the capsid there. So positive sense single-stranded RNA. This virus could just use that to be read at the ribosomes, but it actually doesn't do that. It goes through a lot of trouble. It goes through a much more complicated process. It brings along a copy of that reverse transcriptase. Okay, and you can see that here. And this picture over here is a little bit more complicated than what we need, but they note that there. So what reverse transcriptase does, it takes that single-stranded RNA and uses that as a template to make single-stranded DNA. So if this is plus sense, this would be negative sense, and uses that to make double-stranded DNA. All right, so you can imagine when this was first discovered in like the 70s or early 80s, this is really mind-blowing because this is something that we never do in our cells, right? We never make DNA from an RNA template. All right, so why do they do that? Why does this virus do that? Well, double-stranded DNA is much more stable, and then it can integrate into the host cell chromosome. So that's one of the key enzymes that brings along with it. Another one is called integrase. It's a little more obvious what that does. That enzyme helps that viral DNA integrate into our chromosomes. We'll see a picture of that in a minute. And lastly, protease enzyme. This is necessary for maturation of the virus. Let's say virion. Okay. There's some kind of assembly or maybe some kind of trimming up that needs to be done before that virion is able to infect other host cells. Okay, and a couple of key glycoproteins on the surface of the virion, that GP120 necessary to attach to the host cell. Again, we're going to see this in a graphic in just a second. And GP41, these two helps the virion fuse to the host cell. Okay, don't worry about these other things that they talk about there. Okay, so here's your graphic from your book that kind of walks through the replication cycle of HIV. All right, so here we have HIV attaching with the help of that GP120. You can see the receptor on the host cell is CD4, that CD4 glycoprotein. So what kind of cells have that CD4 receptor? Okay, that's T helper cells. Now, actually, there are other kinds of cells that have that CD4 receptor, which we didn't talk about before, just to not complicate things, but macrophages, for example, have that receptor. So HIV can also infect macrophages, but it doesn't seem to harm macrophages. Okay, it's the T helper cells that end up dying as a result of HIV infection. Of course, that's a huge problem because we know T helper cells are kind of mediating the entire adaptive immune response. So you don't have those T helper cells, you can't get cytotoxic T cell activation, you can't get B cell activation. All right, so it's entering into the host cell here. We get the uncoating of the membrane and the capsid, and right away that reverse transcriptase starts using that sense single-stranded RNA into double-stranded DNA. The DNA, the viral DNA, enters into the host cell nucleus, and with the help of that second enzyme, integrase, integrates into the host DNA, the host chromosome. Okay, so now you can see here they call it a provirus. Remember back our kind of viral terminology when a bacteriophage was infecting a bacteria and it integrated in that lysogenic replication cycle, we called it a prophage. Okay, and we said when that happens with animal cells, we call it latency, and then we call this a provirus. When the virus is integrated into the host DNA, okay, it starts transcribing viral messenger RNA, things necessary for viral proteins, translating them, packaging them up into the virion, and then we get release of the virion. Now, at some point there, that protease function is necessary. And some sources say that happens after the virion is released. There's some trimming or some cuts that make that virion able to infect other cells. Some have that happening still inside of the cell. Whenever it happens, there's something that that has to happen to make that mature virion. All right, how is HIV transmitted? By very specific ways. All right, it's a bloodborne pathogen, so we know by blood transfusions it can be passed by sexual intercourse, IV drug users, so dirty needles, sharing needles, also from mother to child. So about a third of babies born to HIV positive mothers become HIV positive. So it's not necessarily will happen, but definitely a strong possibility. Also, breastfeeding can pass the infection. It doesn't survive long live in the environment. Again, membrane-bound virus, really wimpy in the environment. I can remember, I was old enough to remember when HIV first started making the news, and people worried about, oh, can I get it from sitting on a public restroom toilet seat or a public swimming pool? No. All right, we're not going to breathe it in. We're not going to get it from some kind of fomite. Okay, the year that I could get the data from, I couldn't find anything more up to date in terms of how many people are living with HIV. 37 million infected worldwide, so that's about 1 million in the US. much more prevalent in sub-Saharan Africa. It's estimated that as much as like 25% of the people are HIV positive, again, in some sub-Saharan countries. So 5% of adults in sub-Saharan Africa living with HIV. So that's been a huge scourge there. But, and again, I couldn't find anything more up to date. As of 2011, life expectancy of someone with HIV, 70 years. So we've come a long way. It's not an immediate death sentence like it used to be. All right, so we're getting a lot better at treating HIV. What kind of drugs do we have? And when I updated these slides, I didn't really indent this correctly, so I'll draw some attention to that. Really, we try to inhibit those 3 enzymes that are unique to the virus. So we try to inhibit reverse transcriptase or integrase or protease, right? Like I said, I didn't really indent those quite right. About half of our drugs are reverse transcriptase inhibitors. So it's really common, and usually people, you've heard maybe of the AIDS cocktail. They're taking at least several different drugs. All of these drugs, they're meant to tamp down replication of the virion as well as keep a person from transmitting it. Okay, so they go on these drugs. As soon as they quit taking those drugs, then the virus is going to start replicating again. All right, can you hear me now? Yes, okay, good. So, don't know what I bumped there, sorry. So AZT has a thymidine, and then it's enough to kind of trick the virion into incorporating that into its DNA when it's replicating. Are they still? I don't think so. Did it freeze up? Oh, okay. Let's hope that's okay. Thanks. Okay, so it kind of tricks the virion into adding on the drug into its DNA, but it's not a real nucleotide, so replication will stop or will not be normal. Okay, so the viral polymerases are usually much less discriminate, like they will add anything in there and don't correct their errors very well. So we get really high mutation rate. There's other reverse transcriptase inhibitors that they're not nucleoside analogs, they just bind to the enzyme to, whether it's at the active site or some other site to keep it from working. All right, and then of course, integrase inhibitors try to cut down on the integration of that viral DNA into our genome, and protease inhibitors, again, make it so those virions are not able to go and infect other cells. Okay, all of these drugs have side effects. So normally when a patient will start taking the drugs, they experience some nausea, fever, tiredness, dizzy, they can't sleep. Typically, those early kind of symptoms kind of get less and less, luckily, the longer somebody is on it. But there's long-term side effects like that would fill up pages and pages of possible side effects. So, cancer, like almost anything you can think of are possible long-term side effects. So that's why, the life expectancy is much longer than it used to be, but there are things that can kind of add up there. And I think it's about $20,000 a year. That might be an old number. But so again, you got to stay on the drugs or else, you know, the HIV is going to start replicating again. Okay. So just the pathogenesis, this is a nice figure that's in your book here that we'll parse out a little bit. So if you look at, start right here with the primary infection. Okay, so a patient is infected. So what happens as a response of that, the HIV, I'm frozen again. Okay. Restart screen sharing. All right, let's try again. Thanks for letting me know. Oh, I don't know if it's gonna work. I might have lost everybody down there. I don't know. Oh, all right. Now I've got feedback again, so I must. Let's see here. Okay. All right. We're gonna make it through this semester. We're gonna make it. Okay. How much time we got? We got a little time left. Okay. All right. So, person is infected. All right. So virion is, you know, infecting primarily those T helper cells. So that CD4 helper count, that's the green line, plunges right away. But still have some helper cells, and the body's able to mount a little bit of an antibody reaction. So that's the purple line here. It's kind of going up. HIV will kind of, you know, we get a bunch of it at the beginning, but then as the antibodies rise up, then the antibody titer rises, then the HIV is going to go down. All right, but it's latent. It's still in the cells. You might have a very variable amount of time. It might be years. It might be months when that HIV will kind of reactivate. And so slowly over time, you get the replication of HIV, and at the same time, you get, you know, less CD4 helper cells. In the ability of the body to make antibodies is going down as well. Okay, so a person is considered to have full-blown AIDS when they have less than, I think, 200 T helper cells per mil. And so at that point, typically the opportunistic infections start to be kind of overwhelmed. Okay, so that means the patient is starting to get all sorts of diseases that a healthy person with normal immunity wouldn't get. Okay, so death is due to those opportunistic infections. Okay, what are some of those opportunistic infections? I didn't post this because a lot of these, some of these you haven't heard of before, but this is from the CDC webpage. If a person has, I think if you have HIV and one or more of these opportunistic infections, you're considered to have AIDS, no matter what your CD4 T helper count is. Okay. So again, some of these we've talked about, like here's HHV8, Kaposi's sarcoma. That was that lots of tumors showing up. It's a basically attacking a tumor suppression pathway that we have. Herpes infections that are chronic. You know, pneumonia, we get some weird mycobacteriums that people don't normally get. So again, it's the HIV is causing death indirectly through opportunistic infections. All right, let's see. questions about HIV? All right, so that brings us to coronavirus. All right. I didn't really update this information all that much from what I taught last semester. So I don't really have anything like cutting edge or new here to talk about. Coronavirus, we've known about coronavirus for a long time. It's the second leading cause of common colds behind rhinovirus. All right, so you've probably had lots of coronavirus infections in your lifetime. Of course, this epidemic that we're dealing with is a little bit different. We're going to look at kind of the historical perspective here in a minute. But the structure of the virus, it's a positive sense RNA virus. All right, so, okay, and it is enveloped, right? So all of our viruses that we've talked about, lots of human viruses are enveloped. So some examples, SARS coronavirus, that stands for severe acute respiratory syndrome. That is an infection that showed up 2002, 2003 in China. We're going to talk about that a little bit more. MERS-CoV, that stands for Middle East respiratory syndrome. A little bit more fatal, but hasn't really moved out of the Middle East very far. And then, of course, SARS-CoV-2, that's the name of the virus. When we sequenced it a little more than a year ago, we found that it was really close, quite homologous to the SARS-CoV, so that's why we call it SARS-CoV-2. Different, little different symptoms. Infection looks a little bit different, but again, the structure of the genome is very similar to that SARS-CoV. And then the infection that we get, we've named COVID-19. All right, so again, SARS-CoV-2 is the virus, COVID-19 is the disease. It's a little bit confusing, all the naming. All right, so if you look at the structure of the virion, I got a couple different pictures to show you here. One is a little bit more complex than the other one. That genome is unsegmented. Okay, so you can see here, it almost looks like, I don't know, I was going to say a phone cord, but you guys won't know what that is, like a garden hose maybe. So all this, you know, around here on the inside, that's the genome. It has the longest, actually it has the longest genome of any RNA virus. Okay, and it actually encodes for, I think, 29 proteins. So that's pretty big for an RNA virus. Again, not as big as the DNA virus, herpes virus that we talked about. Okay, and it's unsegmented, so it's all in one piece. You can see that actually maybe a little bit better over here. What's the significance of having a segmented genome versus an unsegmented? For a virus that has polymerases that make a lot of mistakes, it might make more sense to have a segmented genome, because then when it's transcribing each one of those pieces, or transcribing each one of those pieces individually, if it makes a mistake, then maybe a couple of those proteins aren't functional rather than the whole entire strand of it. So again, the polymerases do make a lot of mistakes. It has a helical capsid. So these little orange, they look like little beads on there. That's supposed to symbolize those protein capsomeres that are protecting that RNA genome. It either has 4 or 5 structural proteins. Most sources say 4, so they show 4 on this picture over here: membrane, envelope, spike, nucleocapsid. This picture shows 5. All right, so we're just going to worry about one of those, and that's the spike protein. Okay, because that's the most famous one. That's what we think is the most antigenic. So that's what all the vaccines are trying to, we're trying to make antibodies, make memory cells that recognize that spike protein. Okay, and that the virus needs that for fusion and attachment to host cells. Okay, it's a particular kind of receptor on host cells that it's looking for, kind of a similar function to hemagglutinin in influenza. All right, reservoirs. This got a lot of attention a year ago. Where did this infection come from? Well, we know that all coronaviruses are zoonotic. So there's lots of coronavirus in all different kinds of animals. I attempted to list some of them there. And we know that occasionally, maybe not commonly, but occasionally those can jump species. So, at some point, some coronavirus that was, we think in a bat, at some point acquired the ability to be transmissible to humans, all right? We don't think that's something that obviously happens all the time, but there was some kind of mutation that made that happen. And of course, in Wuhan, where you have wet markets, a wet market is where they sell like fruits and vegetables, dead animals, as well as live animals. So these wet markets got a lot of attention because you have live animals in cages, often in very unsanitary conditions. So you can kind of see in this picture, there's probably about 20 turtles in that little tiny cage, just all on top of each other. Those are live turtles, all right? In some cases, the cages are stacked up, so you have the animals on the top, defecating on the animals below. So, this is just sort of what they do over there. They put restrictions on certain kinds of animals, but it's what people follow those restrictions? It's not really clear. In some areas of China, it's considered a status symbol to eat exotic animals. So the rarer the animal, the higher the status it is if you eat that animal. So that's why you have like all kinds of animals that you've never heard of in these wet markets. So all coming together, it's just a good breeding ground for all sorts of infections. All right, so if we look at the SARS, severe acute respiratory syndrome, whole historical perspective here, that infection killed, there's about 8,000 cases. We don't see this infection anymore. All right, so it lasted for a couple of years and we haven't had any cases since 2003. All right, so the mortality rate here was about 9%. So much higher than what we're dealing with now. A lot of the same kind of similar symptoms. So people died by respiratory distress. Okay. Reservoir here is our bats. It's called the lesser horseshoe bat. Okay, so here's a picture of a lesser horseshoe bat. It's just a little tiny bat, could fit in the palm of your hand. Bats are reservoirs. Remember, a reservoir is oftentimes an animal, but it could be the soil or it could be water where some pathogen is maintained. All right, so we think there's SARS coronavirus, perhaps in the bat population. Bats serve as reservoirs for all sorts of viruses. Why? Well, they live communally. So they live together in caves, just all on top of each other where they're sleeping all day. And they go out at night and travel long distances. And then they come back. So they travel a lot more than any other mammal would because they fly. All right. We think that there is actually a secondary, or what we call an intermediate reservoir, and that's civet cats. Okay, so I included a picture there because that's not something that's familiar to us. but we think that this is kind of an amplifying reservoir. So the cats come in contact with the bats, and then the virus multiplies in the cats. And these civet cats are very commonly eaten in China. Yeah, they're even farmed for human consumption. Symptoms of SARS, kind of the same, a lot of similarities to what we see with coronavirus, COVID-19, fever, dry cough, headache, difficulty breathing, death typically due to respiratory failure. All right, so pretty severe indeed disease, mostly in China. There were outbreaks like in Toronto because somebody got on an airplane and had an outbreak there. But it was only passed when people were ill. So it wasn't transmissible before people were contagious. So we were able to, and it probably wasn't as transmissible as COVID-19. So we were able to kind of put a stop to it. Okay, lots of healthcare workers contracted it as well. All right, and then the Middle East respiratory syndrome, MERS, that emerged about 10 years later in 2012, mostly confined to Saudi Arabia, a few other countries in the Middle East, 858 deaths, higher case fatality rate though, about 35%. Okay, so this is not as transmissible, but definitely more deadly. And we're still having cases of this from time to time. The reservoir we think for this is bats as well. And then there's an amplifying host or an intermediate host of Drominary Camels. Okay, so there's two different kinds of camels. The Bactrian Camels have the 2 humps and the Drominary Camels have the 1 hump. So who's getting MERS? It's people that are in close contact with the Drominary Camels. All right, so human to human transmissible is possible, but not very common. So it's mostly healthcare workers that are treating somebody with MERS that would get it from another human. All right, so to cut down on this, they really tried to focus on people that are in close contact with camels. So camel handlers have to wear personal protective equipment. There have been rules put in place about consuming camel milk and also consuming camel urine, right? If you don't believe me, here's a little article about that. Apparently that is a cultural thing, consuming camel urine. Okay, now that's a no-no. All right, so symptoms, a little more systemic, affecting the kidneys, lining around the heart. Just like SARS, there's no vaccine. But hopefully we'll get better at treating that with all this coronavirus stuff, COVID stuff that's going on. All right, so that leads us to our present pandemic. You know the signs and symptoms of COVID-19. I don't think I need to go through that very carefully. You assess for it every day, hopefully. That loss of taste and smell is kind of unique. That's possible with any respiratory virus because it's killing, you know, your cells in your respiratory tract, your nose. A lot of your taste is kind of centered in your nose there. Extreme tiredness. Of course, there are mild cases. There are severe cases. Death can be due to septic shock or just multiple organ failure, respiratory failure. There are comorbidities, meaning if you have certain health conditions, you're more likely to have severe disease. If you're obese, if you have hypertension or heart disease, you are more at risk. We know that transmission is by respiratory droplets. So typically you're in close contact with somebody else, but it's potential to be airborne. So that's more than 6 feet, more than a meter is considered airborne. All right, so really important to wear those masks to just cut down on that droplet transmission, less commonly by fomites. So, you know, we're using the hand sanitizer and spraying surfaces down just out of an abundance of caution, but we really don't think that's a common way that COVID gets transmitted through touching things, because again, it's membrane, bound virus, enveloped, it's not going to be viable for very long on surfaces. Okay, reservoir. So we think it's probably bats. That'll take a while to determine that for sure. A possible intermediate host, because that first patient in Wuhan had close contact with a pangolin. All right, so I put a picture of that. It's kind of a scaly reptile, not a reptile. It's a scale. It has scales, kind of like a reptile, but it's more like kind of an anteater. They eat insects. Pangolins are actually endangered, but again, the more endangered something is, then the more valuable they are. People eat these in China, so that's a potential intermediate host. We already talked about vaccines quite a bit. The whole Johnson & Johnson thing, it'll be interesting to see what happens with that. But, that clotting is in less than 1 in a million people that took the vaccine. So you're more likely, if you got COVID, you're more likely to die than if you had the vaccine. So I don't know, it'll be interesting what they decide with that. In terms of treatment, there's not a lot out there. Of course, kind of an anti-inflammatory like acetaminophen, people that are hospitalized get supplemented oxygen, or maybe, even some sound like they're doing mechanical ventilation as much now, but that's still a tool out there. Harvesting antibodies from somebody that's recovered. So we call it convalescent plasma. I think the jury is still out on whether that is helpful. It's certainly not harmful, so we're still trying that in some cases. And these are 2 drugs. If any of you caught, I just happened to hear it because I was in the car, the governor's address like a week or 2 ago. She was really pushing the use of this remdesivir to treat COVID. This is another nucleoside analog. So like acyclovir, that has been used for other viral infections like Ebola. So we know that it's safe. So that's really the first consideration, is it's safe. So nucleoside analog, it kind of truncates the replication of the RNA replication of the virus. I don't know. Some studies have shown people to recover faster when they take remdesivir. Dexamethasone, this is a corticosteroid. Okay, and any kind of steroid is going to dampen inflammation. So we know some patients, especially younger people that get COVID, can have a just super, they call it a cytokine storm. So overwhelming inflammation that can actually, lead to death. So in some cases, it's important to dampen that too much inflammation, and so it might be appropriate to give a steroid. That's pretty much what we have now to treat it. I'm sure we'll know a lot more a year from now, but that's what I got for you. That's pretty much all I have to say. We talked about the vaccine a lot already. If you have any questions, you're welcome to hang around. Oh, we only got 5 minutes left. That wasn't too bad. Okay, so next Monday, right here at noon, be in contact with me sooner than later. If you have some kind of conflict, we'll see what we can work out. Okay, otherwise, have a good weekend. We'll see you Monday.